So in the previous lectures, we have discussed about enzymes, their introduction and classification of enzymes. Now we move forward to factors that affect the rate of enzyme catalyzed reactions. Since which are already told that enzymes and substrate, they combine together to form an enzyme substrate complex. But what are the factors that affect these interactions? So there are 5 different factors which regulate the activity of an enzyme. Those being enzyme concentration, concentration of substrate, concentration of the coenzyme, temperature, and pH. An enzyme catalyzes a reaction by forming the enzyme substrate complex, which dissociates So an enzyme catalyzes a reaction by forming the enzyme substrate complex, which dissociates into the enzyme and product. The enzyme may be considered to take part in the reaction. The enzyme may be considered to take part in the reaction as a reactant, though it is regenerated in its original form at the end of the reaction. The rate of initial reaction leading to the formation of enzyme substrate complex is directly proportional to the product of molar concentration of enzyme and substrate. But this simply means that rate of formation of complex is directly proportional to the product of concentration of enzymes and substrate. Similarly, the rate of second reaction leading to the formation of product and enzyme is directly proportional to the concentration of enzyme substrate complex. This simply means that the enzyme substrate complex, the amount of enzyme substrate complex formed will be directly proportional to the amount of enzyme and substrate present for the reaction. And similarly, the amount of product formed and the amount of enzyme left after the product formation is directly proportional to the substrate concentration. Therefore, the rate of formation of product, the rate of overall reaction is proportional to the enzyme concentration, provided that enough substrate is available to combine with the enzyme. The next is substrate concentration. Just as the rate of reaction is proportional to the enzyme concentration, theoretically it should be proportional to substrate concentration also, provided that enough enzyme is available to bind to the substrate. However, the availability of enzymes in the cells is limited, whereas the concentration of substrate can vary over a wide range. When the substrate concentration rises, initially there is a proportionate increase in the velocity of reaction, the rate of reaction. But later the rise in velocity becomes slower until a maximum velocity known as Vmax is reached. As you can see in this graph here, initially the velocity, which this plot is against velocity versus substrate, you can see when the substrate concentration is increasing, the rate of reaction or the velocity is increasing. And it reaches to an ultimate point that is known as Vmax. The velocity is maximum and further does not increase from this point. At Vmax, all the enzyme molecules are saturated with the substrate and no further increase in the velocity is possible, even if the substrate concentration goes on increasing. The substrate concentration at which the velocity is half of Vmax is known as Michaelis-Menten constant or Michaelis constant. This is known as Km value of the enzyme. So Km value is half of Vmax. The relationship between the velocity of reaction and the substrate concentration can be expressed by this equation given by Michaelis and Menten, and this is known as their name, Michaelis and Menten equation. This simply states that the velocity is directly proportional to or equal to Vmax, that is the velocity of the reaction, the maximum velocity of the reaction multiplied by the substrate divided by the Km value plus concentration of substrate. When the substrate concentration is very low, the sum of Km and substrate is nearly equal to Km as substrate is negligible. Therefore, the equation may be rewritten as V is equal to Vmax multiplied by S divided by Km. Since both Vmax and Km are constant, so we can directly say that the velocity is directly proportional to the concentration of substrate. When the substrate concentration is very high, the sum of Km and S is nearly equal to substrate concentration, as Km is relatively negligible. Therefore, the reaction may be rewritten as V is equal to Vmax multiplied by substrate concentration divided by substrate concentration, or we can write V is directly proportional to Vmax. When the substrate concentration is exactly equal to Km, the sum of Km and S may be taken as 2S. So the equation can be written as V is equal to Vmax by half. Thus, when the substrate concentration is equal to Km, the velocity is half of Vmax. Moving on to the next topic, that is the concentration of coenzyme. If a coenzyme is required in the reaction, the concentration of coenzyme can also affect the velocity of the reaction. Basically, coenzyme is a part of the whole enzyme, and that coenzyme can be of two types. It is basically an organic part of that proteinous enzyme. Some coenzymes are very tightly bound to the coenzyme or the main enzyme and form an integral part of the whole enzyme molecule. Other coenzymes act as co-substrate in the reaction. If the coenzyme is an integral part of the enzyme, the effect of coenzyme concentration will be identical to that of enzyme concentration. If the coenzyme acts as a second substrate, the effect of coenzyme concentration is similar to that of substrate concentration. Moving on to the next topic, which is temperature. The effect of temperature is basically initially it was thought to that if temperature is increased, then the rate of reaction or enzyme interaction will also increase. So if the velocity of a reaction is measured at different temperatures, a curve is plotted between velocity and temperature, and we get a bell-shaped curve for that. Initially, when the temperature rises, the velocity increases due to increase in kinetic energy of the reactants. A further rise in temperature leads to progressively denaturation of protein or the enzyme and protein in the enzyme, and the velocity begins to decrease until the reaction practically stops when the enzyme is completely denatured. The temperature at which the velocity is highest is known as optimum temperature of the enzyme. Basically, we get this bell-shaped curve. This is plotted between the velocity and the temperature. Initially, when the temperature increases, the rate of reaction of the velocity increases and it reaches to a maximum point where the velocity is maximum, and that temperature is known as an optimum temperature. And if we further increase the temperature, then the velocity again started to decrease and practically it stops increasing at a certain temperature. So we can say that every enzyme has an optimum temperature at which it performs its best. This is the temperature at which an enzyme activity is maximum. Similar is with pH. If the velocity of reaction is determined at different pH levels and the velocity is plotted as a function of pH, a bell-shaped curve is obtained. A change in pH alters the electric charges on the enzyme molecule and often on the substrate molecule as well. This may affect the binding of substrate to the enzyme or the catalytic activity of enzyme or both. Again, we get the same when we plot a graph between velocity and pH. The pH initially increase in pH leads to exponential increase in the velocity up to the point where it reaches its maximum, and then again further increase in pH will lead to practically stoppage in the velocity or zero velocity at a particular pH. So we can say that initial increase in pH and then further increase in pH leads to formation of this uh, bell-shaped graph where at an optimum pH the enzyme activity is maximum. This is similar to that of our temperature graph. At optimum pH, the velocity of reaction is the highest as the electrical charge on the enzyme and substrate are most suitable for enzyme substrate binding and catalysis. As we move away from the optimum pH, the velocity of reaction decreases. At extremely low or high pH, the enzyme may be denatured, and the optimum pH is different for different enzymes. So we can say different enzyme or specific enzymes, they show particular specificity for pH also. There are certain pH at which an enzyme will perform its best. The next topic is enzyme inhibition. The catalytic activity of some enzyme can be inhibited by certain compounds. Inhibition simply means that the stoppage in the catalytic activity of enzyme. Enzyme inhibition is of two types, basically, the competitive inhibition and non-competitive inhibition. A competitive inhibition is an inhibition in which the substrate and the inhibitor, they compete together. They have a competitive nature towards each other because the substrate, the inhibitor is an analog of substrate. This simply means the inhibitor has a close structural resemblance to the substrate. So the enzyme get confused as to which molecule it should bind. It should bind with its substrate or it should bind with the inhibitor, which is structurally resembling the substrate. Therefore, the inhibitor may be bind to the substrate, the site of enzyme forming enzyme inhibitor complex. We can see in this diagram also here. So that is the diagram. You can see here, this is a substrate and it's binding to the enzyme. These two form the enzyme substrate complex and product is released along with the enzyme. Now, at the same site, an inhibitor can bind. And if an inhibitor binds with an enzyme, it forms an inhibitor enzyme complex, but that will not lead to the formation of the product. So this type of inhibition is known as competitive inhibition. Both enzyme substrate and enzyme inhibitor complexes will be formed, and but only the enzyme substrate can form the product. We can see here that if three things are present, the enzyme, the substrate, and inhibitor in a solution or in an area, this will lead to the formation of two complexes. Some enzymes will bind with substrate to form ES, and some enzymes will form EI with inhibitor. Now only the product will be formed from enzyme substrate. As you can see here, an enzyme inhibitor will block the reaction of the activity of the enzyme. There are certain competitive inhibitors which are used as drug. As we know, there are disorders where enzyme activity is increased. So to inhibit the enzyme activity and to cure the disorder or to get relief from the disorder, there are certain drugs which resemble the enzyme substrate, and they just basically block the enzyme and lead the reaction to get stopped. So these are allopurinol, phytostigmine, mavastatin, lovastatin, and aminopterin. We will go one by one. The first one is methopterin and aminopterin. These two resemble folic acid in structure. You can see here the structure of folic acid is here, and aminopterin is there. So these are the competitive inhibitors of dihydrofolate reductase, DHFR. And here folate and NADPH react to form dihydrofolate in the presence of dihydrofolate reductase. This is the enzyme. And this dihydrofolate again further reduces to form a tetrahydrofolate along with NADP. This reaction is important in the synthesis of purine and thymine nucleotides. Inhibition of this reaction decreases the availability of nucleotide for DNA synthesis and inhibit the cell division. So we can see if we want to decrease cell division, that's generally the case in cancer. Therefore, these two are used as anti-cancer drugs to suppress the unregulated cell division that occurs in cancer. Next one is allopurinol. It is one of the famous drugs and of enzyme competitive inhibitors. Basically, allopurinol is a structural analog of hypoxanthine, and allopurinol you can see here. It is a competitive inhibitor of xanthine oxidase. So allopurinol resembles hypoxanthine structure, and xanthine oxidase converts hypoxanthine into xanthine and xanthine into uric acid. So in gout, we know that there is collection of uric acid because the production of uric acid is too much. So by using allopurinol, what we are doing is that we are blocking xanthine oxidase enzyme, and thus we are blocking the production of uric acid. These uric acid, basically what happens that when it is formed, it forms crystals and it gets deposited in the joints and leads to excruciating joint pains. In gout, which results from overproduction of uric acid, allopurinol is used to inhibit the formation of uric acid. Next is phytostigmine and neostigmine. These resemble acetylcholine in structure and are competitive inhibitor of acetylcholinesterase. Acetylcholine and water reacts to form acetate and choline. They decrease the breakdown of acetylcholine. Acetylcholine is a neurotransmitter, as we all know. They are used as drug in condition in which the concentration of acetylcholine needs to be increased. Myasthenia gravis, an autoimmune disorder in which acetylcholine receptors are decreased in number. So, in order to increase the acetylcholine concentration, its degradation is stopped by using these two. Next is mivestatin and lovastatin. These two are basically cholesterol or lipid controlling drugs. These resemble HMG-CoA in structure, and they are competitive inhibitor of HMG-CoA reductase. HMG-CoA reductase catalyzes a reaction which leads to the formation of mevalonate and ultimately cholesterol from HMG-CoA. This is a key reaction in the synthesis of cholesterol. Inhibition of this reaction decreases the synthesis of cholesterol. Therefore, mivestatin and lovastatin are used as hypocholesteremic drugs as they stop the formation of cholesterol. A non-competitive inhibition is a type of inhibition where there is no structural resemblance between the substrate and the inhibitor. So they do not compete with the substrate for binding to the enzyme. They bind to some other region of the enzyme and renders it inactive. Non-competitive inhibitors, non-competitive inhibition may be reversible or irreversible. Generally, it is irreversible. Examples of non-competitive inhibitor include heavy metals, iodocetamide, and p-chloromercurobenzoate.